This video I've prepared for the grade 12s to help them with their June exam preparation. In this video, we'll be looking at the KZN June 2020 exam paper. Before you watch the video, make sure you have the memo and the question paper at hand. Right, 1.2. Give the correct biological term for each of the following descriptions. Write down only the term next to the question number in the answer book. Right, 1.1. All the genes that make up an organism, we should know that that is a genome. 1.2. An inherited disorder where blood fails to clot properly, that we know is hemophilia. Take a note of the spelling of the word hemophilia. 1.3. An alternative form of a gene that has at the same locus, that we know is an allele. 1.4. The membrane that together with the endometrium forms the placenta, that we know the membrane is the chorion. Again, note the spelling. 1.5. The structure in the head of the sperm that contains digestive enzymes to penetrate the ovum, acrosome. 1.6. Nerves linking receptor and effector organs with the brain and the spinal cord, that will obviously be peripheral nerves. 1.7. The formation of gametes by meiosis, gametogenesis. 1.8. The hormone that is produced by the corpus luteum to further thicken the endometrium, that we know, progesterone. And 1.9. The part of the amniotic egg which stores waste, allantoin. Right, 1.3. Indicate whether each of the descriptions in column 1 applies to A only, B only, both A and B, or none of the items in column 2. Write A only, B only, both A and B, or none next to the question number in the answer book. Right, 1.3.1. What condition affects the lens? A condition affecting the lens of the eye. Now, what condition affects the lens of the eye? Of course, we know it's a cataract. Astigmatism affects the cornea, so your answer is B only. 1.3.2. The outer jelly-like layer of an ovum is not the cytoplasm and it's not the shell, so your answer is none. 1.3.3. A disorder caused by the degeneration of the myelin sheath of motor neurons, that we know is multiple sclerosis and not Alzheimer's, so your answer is A only. 1.3.4. The innermost membrane surrounding the fetus, that we know is the amnion and not the cell membrane, so your answer is A only. And 1.3.5. The scientist who discovered the structure of the DNA is James Watson and Francis Crick, so your answer is both A and B. 1.4. The diagram below shows one pair of homologous chromosomes found in each of 4 butterflies. The alleles for antenna shape, normal or abnormal, and body pattern with stripes or without stripes are indicated on the chromosomes for each butterfly. Here's it here in the key. So, 1.4.1. Write down the number only of a butterfly that is heterozygous for body pattern only. Now, body pattern only. Let's look at body pattern. We're looking at the S and this is the stripes. So, body pattern only, heterozygous for body pattern only. My answer is 4. Right. Write down the number only of a butterfly that is homozygous for both characteristics. So, homozygous for both characteristics, as you can see, it's in butterfly 1. Right, 1.4.2. Write down the phenotype for butterfly 2. Now, butterfly 2, can you see? It has body with stripes or striped body. And can you see? It has normal antenna. As you can see for using the key. 1.4.3. What will the phenotypic ratio of offsprings? What will be a phenotypic ratio of offsprings if butterfly 3 was crossed with a butterfly with the same genotype as butterfly 3? So butterfly 3, can you see, is heterozygous. So heterozygous plus heterozygous. When you have 2 heterozygous butterflies for both characteristics crossed, your ratio will always be 9:3:3:1. So you get 9 striped with normal antenna, 3 striped with abnormal antenna, 3 no striped with normal antenna, and 1 no stripes and normal antenna. Nice question. 1.4.4. Write down all the possible gametes from butterfly 2. Now butterfly 2, I wrote down here, is big S with heterozygous for N, for antenna. So how do you work out your gametes? First, outermost, innermost, and last. And so there's your two gametes, S big N, S small n. Right, 1.5. Study the diagram of a neuron below. 151. Identify the type of neuron shown in the diagram. This we know is a sensory neuron. 152. Identify parts A. Part A is this entire structure with the cell body, or with the nucleus, sorry. That's the cell body. Right. Identify part C. Part C is pointing to that long thread. So that, of course, is the axon. And then 153. Give one function of part B. What's the function of the nucleus? It controls the function of the neuron. Give one function of part C, which is the axon, carries the impulse away from the cell body. And then give one function of D, which is your dendrites, transmits the impulse towards the cell body. Right. 2.1. The diagram below shows protein synthesis, as you can see in the diagram. 2.1.1. Identify organelle V. Organelle V is here. That, of course, is a ribosome. 2.1.1b, molecule Q. Molecule Q, can you see? Obviously, it carries the amino acid, so that's tRNA. 2.1.2, write on the nitrogenous bases for the codon R if it codes for glycine. Now, this is the mRNA. And this is the tRNA. So it says that R codes for glycine. So opposite G will be C, opposite G will be C, and opposite A will be U. So C, CU. Then it says 213. Write on the sequence of the first two amino acids from left to right, which codes for molecule Q. Right. So opposite U here will be A, then UA. Is there one? Yes. What's the amino acid? Isoleucine. Then opposite G will be C, opposite G will be C, and then A, there's your amino acid, CCA, what is it? Proline. Okay, right, 214. Explain the effect on the protein formed if all guanine bases in the molecule Q were replaced by cytosine bases. So, the tRNA will change its base sequence, right? So, we look at Q. If it were all the G's here were replaced by C's, so we know the tRNA will change its base sequence. Proline. So if you look at the picture, Q is GGA. Now they're telling you this GG is replaced by CCA. And so that's why the proline will now be replaced by glycine, because obviously the amino acid that will code for this will be glycine. So, and it will lead to a different protein forming. So, that is a bit difficult to understand, but a very nice question. This is the nice question, 214. Try and figure it out. Write the nitrogenous bases. So it's supposed to be GGA. Then it gets replaced by CCA. And what would have been the amino acid? It would have been proline, but now it will get replaced by glycine. Okay, so amino acids change and a different protein will form. Okay. 215. Tabulate two structural differences between molecule P and Q. So P is a DNA and Q is a RNA, or specifically an mRNA. So P is double-stranded, mRNA is single-stranded, DNA has thymine, RNA has uracil, DNA is a long molecule, and mRNA is a short molecule. Right. Don't forget to make sure to draw the table. 216. Describe the stage of protein synthesis which involves molecule Q. That involves the tRNA, so that process is translation. So, for translation, each tRNA picks up a specific amino acid. When the anticodon on the tRNA matches the codon on the mRNA, then the tRNA brings the required amino acid to the ribosome. Amino acids become attached by peptide bonds to form the required protein. That you will find in the exam guidelines as well. Right, 2.2. Artificial selection or selective breeding is a choosing of parent organisms with a known genotype to produce offsprings with specific traits in quantities within a short space of time than in nature. A farmer wanted to produce sheep that are goodwill and meat producers. He performed the genetic modification and recorded the number of offspring that produced both goodwill and meat. The graph below shows the results of the investigation over 5 generations. So there you have the success rate in percentage generations, success rate of artificial selection over 5 generations. So 221, identify the dependent variable. So the dependent variable, as you can see, is of course success rate. Okay. Right. Identify B, independent variable. What was he controlling? He controlled the generations. 222. What is the percentage rate, a percentage success rate of generation 5? So if you look at it, it's obviously 45%. Right. 223. Explain one way in which the farmer could use the results to his advantage. He would produce sheep with goodwill and also good meat. 224. State 2 ways in which he could have ensured validity in this experiment. He could have used the same species of sheep, and he could have used the sheep of the same age. Give a conclusion for this experiment. As you can see, the amount of wool and meat production increases from generation to generation due to artificial selection. Here it must be in this order, but you can see it in the graph. Okay. 226. Give one reason why the investigation was carried out over 5 generations. Of course, to increase reliability. Right. Next question, 2.3. Auditory processing disorders occur when the brain has problems in processing the information such as understanding speech. Conductive hearing loss occurs when there is a problem with the outer or middle ear, which interferes with passing sound to the inner ear. Sensory neuron hearing loss occurs when some parts of the ear are damaged or malfunctions, so it is unable to accurately send the electrical information to the brain. The table below shows the percentage of people who suffered from different types of hearing loss. So here you have your types of hearing loss: auditory processing, conductive, sensory neural, and mixed. And there's the percentage of people suffering. 231. Name the part of the brain that is damaged if the person suffers from auditory processing disorder. That's easy, cerebrum. 232. Name 2 parts of an ear that are most likely damaged in a person who has sensory neural hearing loss. That, of course, is the cochlea or auditory nerve. 233. Explain why conductive hearing loss may be temporal than the other types of hearing loss. Because it is the earwax that blocks the ear, and that can be removed, and the structures that conduct hearing can heal over time. Then 234. Draw a bar graph to represent the information in the table. So there you have graph showing the percentage of people who suffer from the different types of hearing loss. You have the percentage people suffering in percentage and the types of hearing loss. There's your bars. Your spaces must be the same width, and the width of the bars must be the same. So remember, the width of the bars must be the same as well as the spaces. Right. 3.1. Read the extract and study the diagram below. An undescended testicle that hasn't moved into the scrotum. An undescended testicle is common among baby boys born prematurely, born of smokers, alcohol drinking, or an alcohol drinking mother. The undescended testicle moves into the proper position on its own, but if not, surgery can relocate the testicle into the scrotum. This may lead to infertility in men. 311. Identify part C. Part C is the coiled part next to the testes, that is the epididymis. Identify part E, that, of course, part E is the seminal vesicle. Here I've written it down here below. 312. Name 2 risk factors from the passage that may cause undescended testicle in male babies. Of course, this is going to be mothers who smoke while pregnant, mothers who drink alcohol while pregnant, and premature birth. 313. State one function of part B. Part B is the urethra, so it transports semen to the exterior. Right. 314. Explain how undescended testicle may lead to infertility in men. So the testicle remains inside the body and receiving temperature equal to the body. So the body temperature and the testes are the same, and this will affect sperm cells, leading to a decreased sperm count. Because remember, the body temperature is too high for spermatogenesis. The testes has to be 2 degrees below body temperature. Right. 315. State 2 pieces of advice you would give to a pregnant woman to eliminate the risk of undescended testicle tube in their child. Tell them, do not smoke while you're pregnant and do not drink alcohol while pregnant. Right. 316. Explain the consequence if Part D was surgically cut and tied. Part D is the vas deferens. What will happen if it is cut or tied? The passage for sperm to reach the urethra will be blocked. No sperm will enter the urethra. No sperm will be present in the semen. No sperm to fertilize the ovum. No pregnancy. Right. 316, that was 316. 317, describe the process by which sperms are formed. That's spermatogenesis. Straight from the exam guidelines. Under the influence of testosterone, diploid cells in the seminiferous tubules of the testes undergo meiosis to form haploid sperm cells. Right. 3.2. Study the diagram of a human eye. 321. Name the process illustrated in the diagram. Here you can see the pupil is changing size. And that is, of course, the pupillary mechanism. 322. Identify parts A. Part A is pointing to this part here, and that is the pupil. Right. Part B is pointing to the white part of our eye, and that we know is the sclera. 323. Which change, COD, occurs in the light when you're watching television and suddenly the power goes out during the night? That, of course, is part D because the pupil will dilate. 324. Describe the change in D. The circular muscles will relax. The radial muscles will contract. The pupil dilates to allow more light to enter the eye. Right. 3.3. The following diagram shows the inheritance of fur color in squirrels. Black color is controlled by the allele big B and white color big W. So here you see the pedigree diagram and the key. 331. From the pedigree diagram above, how many individuals are males with black fur color? So there are only 2, as I've ticked them off, that are black and are males. Then from the pedigree diagram, how many individuals B are heterozygous for fur color? Heterozygous for fur color, as you can see in the key, there's in the pedigree chart, there's 3 of them and 3. Just correct the memo. Your answer had 4, but the answer is actually 3. Right. 332. Name the type of dominance shown in the pedigree diagram. This we know is incomplete dominance. Right. 333. Explain why grey color offspring was produced when mating individuals 1 and 2. When black and white squirrels are mated, they produce a grey offspring, which is a third phenotype, since the alleles for black and white are neither dominant nor recessive. Right. 334. Identify the phenotype for individual 2. That's easy. As you can see, it's white. 335. How many generations are represented in this diagram? 1, 2, 3. So your answer is 3. And then 336. Give the genotype of individual 7. Can you see that person is, this squirrel, is grey. So it's big B and W, which gives us obviously grey. And then 337. Do a genetic cross between individuals 3 and 4, which is a white squirrel, white female squirrel, and a grey male squirrel. So P1 phenotype, white female and grey male. Genotype, big W, and then big B and W. Then you write the words meiosis, fertilization. You have P1, F1, meiosis, fertilization gives you a mark. And look at the gametes. Big B, big W, WW, and then this is your answer. Big B, big W, big B, big W, and big W, big W, big W, big W. So it's 2 white, 2 grey, which is a 50% chance. Make sure you have these marks here. The P1 and F1, meiosis and fertilization, are always easy marks for you to gain. Alright, then we're looking at question 4. Now, I still do question 4, which is an essay, which is not tested anymore, but these questions can be broken down into shorter questions in your exam. So describe how meiosis leads to genetic variation and describe the role of meiosis in oogenesis. Also describe how mutations results in Down syndrome. So let's look at genetic variation. Prophase 1. We're looking at how meiosis leads to genetic variation. Prophase 1. DNA untangles to form visible chromosomes. The nucleolus and the nuclear membrane disappears. Homologous chromosomes come together, forming a homologous pair. Two non-sister chromatids of homologous pair overlap, forming a point called the chiasma, at which genetic material is exchanged during a process called crossing over. Still genetic variation in meiosis, metaphase 1. Random arrangement of chromosomes occur as they line in the equator in pairs, following no pattern, and the chromosomes are held by spindle fibers. Now, they ask us the role of meiosis in oogenesis. The diploid cell inside the follicle or the ovary enlarges and undergoes meiosis. 4 cells are produced. Only one survives to form a mature haploid ovum. And lastly, how mutation results in Down syndrome. During anaphase 1 or 2, chromosomes or chromatid on pair 21 may fail to separate or non-disjunction occurs, resulting in a gamete with an extra chromosome on pair 21. When this gamete fertilizes with a normal gamete with 23 chromosomes, it will result in a zygote with 47 chromosomes, leading to Down syndrome. Right. Thank you, grade 12s. Thank you for watching my video. I hope you learned a lot. Join me and watch all my other videos as well.